of all, I want to thank the organizers for giving me the opportunity to give a talk today, and of course, thank all of you to stay here and listen to me, since it's been quite. We lost a lot of people. Anyhow, so today I want to give you here a look into my PhD thesis, actually. So I'm working with Clostridium perfringens enterotoxin as the mediated suicidal gene therapy for pancreatic cancer. Pancreatic cancer itself is it's a 12th common cancer worldwide, and it's the 7th cause of cancer-related death. And this particular type of carcinoma has a really poor prognosis, with a 5-year survival less than 5%, and median survival between 3 and 6%, or 3 or 6 months. So, which, the molecular heterogeneity, the incomplete knowledge of this disease, and of course the resistance to existing treatments brings the urgent need of the identification and characterization of novel targets, molecules, and also new strategies of treating this cancer. So one of the strategies for us would be the cancer gene therapy, and as you know, you can either target the host or you can target the tumor cell, and we would like to focus on to targeting the tumor cell by killing this cell itself, and we're focusing on direct killing using the suicide gene therapy with the help of bacterial toxins. Talking about bacterial toxins, so they have become more into focus, and they've involved their role within the cancer treatment, and I've listed 4 of them. So the diphtheria toxin and also the Pseudomonas exotoxinae have been used for different tumor entities. So which button is it actually here? So it has been used for glioblastoma and melanoma, and there's also a different group of bacterial toxins, which are pore formation toxins, which perform cell lysis via different targets, as I would name it right now. And we want to focus on the Clostridium perfringens enterotoxin, which acts via the Claudin 3 and 4, which I will introduce later on. So the Clostridium perfringens enterotoxin is produced by the gram-positive bacterium Clostridium perfringens strain A, and as you see here, that's the bacterium, and usually it releases its toxin by sporulation. And this toxin is a 35 kilodalton protein of 319 amino acids. And this toxin has its natural receptors, which are the tight junction proteins, Claudin 3 and 4, which are pictured right here. Why are we looking into tight junction proteins? They have become so they got more into focus, and it's been published, and it's been known that their expression is altered, and so within different cancer entities you can find a different expression of different claudins. So it's a family of 24 proteins, to mention that. So you can find upregulation and downregulation in different epithelial cancers, and we are focusing on pancreatic cancer, so the literature says there's an upregulation of Claudin 3 and 4, which of course helps us, since these are the natural receptors of the CPE. So to show you the action of CPE when it finds its receptors, it's shown here. So this is the CPE, which has a receptor binding domain, which is on the C-terminus, and it also shows a cytotoxicity domain, which is on the N-terminus, and by binding with the C-terminus to the extracellular loop of the claudin, the pore formation starts, and as more CPE, so as more toxin comes closer to its receptors, a big pore formation starts, and the pore is builded, and the cell undergoes cell death. So with this knowledge, we designed the study, and the aim of my study is actually to characterize the pancreatic cancer cell lines. And also further on, we want to, of course, extend our approach and want to look into patient-derived xenografts. Of course, then after characterization, we would like to target our cells with the local treatment of recombinant CPE and also the CPE gene transfer. And then with having this, we, of course, wanted to know and understand the mechanism and the kinetics of the whole approach. with this knowledge, we want to proceed in vivo experiments, and of course for that we need transplantations subcutaneously and octotopically. And the major aim of the study is, of course, to evaluate selective CPE gene therapy for pancreatic cancer overexpressing Claudin 3 and 4. So to present you some of my results, I want to start with the human pancreatic cancer cell line panel, which we, of course, checked for the expression of Claudin 3 and 4. So and as you can see, we performed Western blot, and also we did some immunohistochemistry to mention that. So it's been known in literature that the melanoma cell in SKML5 is not overexpressing any Claudin 3 or 4, and the colorectal cancer cell in HT29 is an overexpressing cell. So we used these two different cancer entities as positive and negative control, and as you can see, we did not detect any of the claudins in the negative control and have the positive control shown here. So what else you can see that the expression of these two claudins is different within this pancreatic cancer cells, and but we do have an expression of either one and or the other claudin. We also performed the immunohistochemistry to, of course, look into how is the distribution of the claudin. So for example, right here you can see that I hope you can see, maybe it's too little. The claudin sits right here more within the cell-cell contact, and on this, for example, the claudin is distributed within the cytoplasm. So the distribution and the expression is different within the panel. So of course we wanted to see if the CPE actually does show its action. So we took the recombinant CPE and treated our cell lines with different concentration, and as you can see, we could clearly see in the HOOP-T3 and the CAP1 cells that there is a concentration-dependent cytotoxic effect. Also with ASPC1, we were able to detect a reduction of the viability, and also within those representative pictures, you can see, like, maybe not really, but cells remain vital and healthy with treating them with with the control, and by using the REC CPE, the cells immediately die, and so this is actually measured 72 hours after treatment. And as you also can see is that the negative cell lines SKML5 remains unaffected. So with this knowledge, we wanted to move on, and of course wanted to do now our gene transfer for anti-tumoral therapy. So for that, we cloned the CPE cDNA from Clostridium perfringens DNA and generated a translational optimized cDNA, which we will call opt-CPE, which is right here. So we took this and transfected this in vitro in our cell line panel, and later on I will show you the in vivo data. So first of all, we wanted to see if this actually works. So for that, I should mention, we also created the opt-CPE and GFP construct, so we just added the GFP to it. So as you can see here, 12 hours after transfection, we were able to detect the CPE. We see the distribution within the cells, and pretty much all the cells are expressing it after 12 hours. We also were able to detect the distribution of the claudins, which are cytoplasmatic and also within the membrane. And as you can see, 14 hours after transfection, the well-organized cells are becoming unorganized, chaotic, and cell death has started. So we also confirmed this with our MTT assay after 72 hours, and as you can see, HOOP-T3, Caprin-1, PATU-1, there pretty much all the cells show a significant reduction of cell viability, and the negative control remained unaffected. You also can see that there is a difference, right? So you see there are cells that are reduced to 20% of viability cells, and there are just some they're still having 70% of viability, which again says that there is a correlation of cytotoxicity, like the cytotoxic effect, that this one correlates with the expression of the claudins, then of course the transfection efficiency, and also the distribution of the claudins really plays a role in this case. What's also been known and has been published before is the bystander effect, which helps, of course, the gene transfer. So to explain it really shortly, so you have the direct killing by bringing the vector into the tumor cell. The tumor cell itself produces the toxin, which gets out of the cell, binds at the membrane, and brings the cell into cell death, but also this released CPE gets into the neighbor cell, which is not transfected, and kills this cell as well. So to check this and prove that if we have this bystander effect or not, we took the supernatant of the negative cell line SKML5, and as you can see, we detected opt CPE concentration 24 and also 48 hours after transfection, and not, of course, with the empty vector. Then we wanted to quantify this CPE, so we did. So as you can see here, we used the CPE ELISA. In 24 hours, there were like 30 nanogram per milliliter detectable, and 48 hours, there was around 430 nanograms detectable. To see whether this released CPE is actually still biological active, we again transfected the HOOP-T3 cells. Of course, we saw again that there's a reduction of the viability, and we collected the supernatants 24, 48, and 72 hours and added that to non-transfected respective cell lines, which you can see here. And just by adding the supernatant, the cells lost their viability. So to say that, this is measured 72 hours later. So, but just with the supernatant, we were able to kill the cells again. So the biological activity has been proven with that. So then, of course, you're always interested what kind of cell death mechanism is going on. And I've done some NXEM-PI measurements, and even so with the immunofluorescence, I was able to see that we have different kind of cell death going on. Most of the times you see the budding, which indicates necrosis. Maybe you can see it right here. But also you can find the blabbing of apoptotic cells. So for that, we kind of did two assays. So we checked the LDH release, which is always indicating necrosis, because if a cell bursts, the LDH is just massively released into the supernatant. And we also checked the caspase activity, which of course gives you an indication of apoptosis is going on or not. So, and as you can see here with the Pahle cells, we have a high amount of released LDH, since it's like around 13. As you can see, even with the empty vector transfected cells don't really show enough, but with the up-transfected cells, we definitely see an increase of the LDH release. You also find it in those two cell lines, but it was not smart of me, but this has a variation to 15, and this is just to 4. So it's definitely less than here. And what you see on this side now is that if you look right here, this is the carbon activity, or the caspase activity of carbon-1 cells. And you can definitely say that there's an activity of these caspases, which can be explained because it's depending on the CPE concentration. If you have a low concentration of the CPE, it leads towards apoptosis, but if you have a high concentration, it goes into necrosis. It's also been published, and I tried to kind of show it right here. So then, of course, having all the in vitro data, we wanted to go in vivo, and for that, first of all, we had to establish a new stable transduced luciferase expressing cell line, and we again used the Pahle cells and transfected those into nude mice, 10 to the power of 7, right there. And then after these tumors were palpable, we started the in vivo measurement, and by the time, so 17 days after transplantation, the cells were big enough to treat them with the jet injection. So we injected 50 microgram of opt-CPE vector, and as you can see here, 24 days after transplantation, there was a visible and really strong reduction of the cell viability in the CPE-treated tumors, and also, of course, 28 days. So concluding out of this, so our tumor viability is reduced after CPE treatment in those animals. And Then we wanted to proceed and extend our approach, so we checked into the patient-derived pancreatic xenografts. And of course, first of all, we characterized our xenografts. So as you can see in those 5 models, we have a strong upregulation of one or the other claudine, and it is also shown in the immunohistochemistry. And too bad that you can't see it, but the distribution is definitely right into the membranous. So then we wanted to check, of course, again, does the CPE actually bind to those PDX? For that, we didn't want to go right away to the animals, so I took the tumors and put them in vitro, so we call those the PDX-derived cell lines. And having those cell lines, I was able to test them again with the recombinant CPE, which is pictured right here. And as you can see, with the high dose of recombinant CPE, we were able to reduce the viability of those tumor cells. And this is absolutely preliminary, but we just wanted to see if we have kinetics, if we have a distribution. So we again did the in vivo gene transfer with the jet injector and took the tumors. And as you can see, 24, 48, and 72 hours, the tumors do express the CPE. And also, you can see it right here. So the CPE accumulates right there and here. And also here, it is also distributed within the tissue. Then we took the tumors, and as you can see, the control-treated animals reveal vital tumor tissue. And right here, I hope you can see it. There's a massive necrotic area within this tumor. Even so, I have to say that we did not see any reduction of the tumor volume itself, but after checking the immunohistochemistry, we definitely saw this result, and having this in mind, we decided we have to create new models or like a new way to detect our effect. So again, we will proceed with patient-derived xenografts cell lines to transduct them with a luciferase construct. So we were able to actually really see the effect by minimizing the viability of the luciferase signal. All right, just to sum up really fast, the gene transfer of Clostridium perfringens leads to efficient expression in tumor cells. It causes intracellular CPE accumulation and release. It induces rapid toxicity in transfected cells, and it's restricted to claudin 3 and 4. It also mediates the bystander effect, and it does induce necrosis, also late apoptosis in tumor cells and tissue, and it leads to inhibition of tumor viability in vivo. So this whole approach could be employed for selective suicide tumor gene therapy and pancreatic cancer, which are overexpressing claudin 3 and 4. All right, with this slide, I just want to thank the whole group I'm working at and my collaboration partners. So EPO provides me with the PDX models. Also, they have Jörg Piontek and Anna Piontek, which help me out with certain constructs. And this whole work is funded by the German Cancer Ed. So, and thank you very much for listening.
So it's not on the cell surface anymore, I would assume. How does CPE enter the cell? I would assume it needs to bind claudins on the cell surface to enter the cell. But in the tumor cells, the majority is not on the cell surface anymore. Is that correct?
Well, since this is still one thing I really have to find out how the CPE, when it is produced by the cell, it somehow gets out of the cell. So right now I have evidence it could be via the exosomes or it's endosomal, but somehow it does bind within the cell. I've seen this, that it also CPE and claudin binds within the cytoplasma. So I still can't really answer this question when I've been working on it a lot, but it also gets out within in the supernatant, it binds back to the cell and binds what? Binds the claudin 3 and 4, which is actually, yeah, it is expressed on the cell membrane. Not always super strong, but mostly you really, I need better images.
Even if you have only slight expression of claudin 3 and 4 on the cell surface in the tight junctions, you will still get efficient entry of CPE into the cell.
Within the tight junction is always hard as well too, because then of course it's hard to get there.
Well, tumors usually probably don't really have a lot of tight junctions.
No,
anymore, right?
exactly, they're altered, so that's where the expression of the claudins is all over the cell or within the cytoplasm. But as I said, it could even be that they bind within the cytoplasm. This is, I know it's not a really good answer, but this is what I see, and this has to be clarified.
I was unclear about what was the vector you were using, and my second question is: did you see toxicity in the animal? But you saw that it was worse on the tumor. Well, what happened to normal animals?
So the animals itself don't show any side effects, and even when we use the empty vector. So in this case, since I used the CPE GFP construct, it's a pcDNA 3.1 vector which has a GFP bound to it. It's just with a kit prepared. So, and we used as control, we used the GFP empty vector and put the empty vector into the control animals, and the
So you were injecting plasmid IV?
Not IV. No. We do it locally. Okay, I should have mentioned that before. No, we're not doing this IV. So this is what we do. So we have the tumor right here, SC, and we have this jet injector which works. So we put the naked DNA pretty much in the head of the handle and use high pressure. So it works like a diabetic pen. So we use high.
So that would work for, I guess, sub-Q model.
It's right. You are absolutely right. It's a local treatment option right now, and it's right for pancreatic. Of course, that won't work in clinic because we will test it. Then we do a needle injection, and then also do electroporation, which could help at the operation site orthotopically, but it's not clear how we will do it. So that definitely needs more work and more thoughts.
How did you check the transcription efficiency of the clone gene?
Oh, excuse me.
How did you check the transcription efficiency in the clone from the clone gene?
I just didn't get the last part.
Of the cloned gene, just the transfection efficiency of the cloned gene when you're transfecting that into the tumor cell.
Oh, this is just so I took the chuck frozen tumors and did some sections and lysate that and put the lysates onto the Western blot. So this is just really taking the injection site and taking a cryostat and taking sections and see whether the CPE is there, and also with this. So pretty much these stained fractions are also used, not the stained fractions, but those kind of fractions are used for the distribution analysis.
Did you get uniform distribution of CPE in the tumor related to this question? Or did you get localized expression?
It's pretty much right now, so it's more localized. So I see it on the injection. So when we use the jet injector, we have 5 injection sites, and I definitely see it on the channels. So since we're using a high pressure, I see it along the shoot channels. So I see the distribution around
So the
there.
bystander effect is not as pronounced in the animals?
As I said, this is very preliminary. We have to do more work in the animals. So and I hope we will see better distribution within the animal. But if I look here, and this is actually like a 5-time magnification, so the whole tumor had the outer membrane. It was viable and it was vital, but inside the treated animals, the tumor was necrotic, like all over. It's like maybe there are a few islands which are still vital, and of course these will at one point reproduce and be more vital again. But this is something we have to clarify.
Sorry, I think I'm not sure if I understood. Did you say the control? It was also expressing CPE, right?
No. The control just expressed the GFP. So I have
Did
the.
you try an experiment where you just injected the CPE in animals that doesn't have any pancreatic cancer?
Well, that could be an option, yeah. But I also have, with the collaboration of the FMP, so they just published that they have a non-binding CPE. I will use that for my.
We need to see if it's actually specific to pancreatic, like if it's just specific to Claudin 3 and 4 that's overly expressed from pancreatic cancer.
Or if the CPE maybe binds to something else.
Yes.
You mean, yeah. But that's why we would use the non-binding CPE construct, which I will get from the FMP. So this is in planning as well. So that could help us.
So just an extension of that question, because of the idea of the toxicity and the efficiency of transfer of the gene once you put it into the tumors, I think you're on the right track. You must have that bystander effect because the efficiency of incorporation of the transgene is never going to be 100%.
Exactly.
So you do rely on that. That's important to have. But where is Claudin 3 and Claudin 4? Where are they expressed in humans? What tissues are they expressed
Or
in?
all epithelial tissue. So of course that's a
all
big.
epithelial tissues.
Yeah,
So that could be this. So you're going to have some off-target toxicity. One assumes in humans.
Exactly.
Yeah. Okay. Now in mouse, it's not going to. The CPE you're using is not going to hit epithelial tissues. In
So
the mouse?
what we did when I did this, we also took the blood 24, 48 and 72 hours, and we were not able to detect any CPE. So
once
it's
the CPE is there and expressed, it's actually going to be quite local.
Yes,
It's unlikely to be. It has a very short half-life, so that's to your advantage too.
exactly. So yeah, we definitely are aware of the cytotoxic effects. So because all the times people ask, so if they work, why don't you go IV? It's like people would suffer. It's really important. Yeah. So that's why we're totally limited on the local
I see.
application, and that's the only point we can do probably if the patient gets surgery, and maybe we can go right in the surgeon side. So that would be right now the only option. Maybe there could be any opportunity to combine that with nanoparticles. That could be an option, but
I see. Okay. Thank you very much.
well, thank you.
Further questions? If not, thank you very much.